So next chapter is the transport of the respiratory gases. As we know that the oxygen that is essential for the cells, it is transported from the alveoli of the lungs to the cells. Whereas the carbon dioxide that is the waste product in the cells, it is transported from cells to the lungs. Now for the MCQ point of view, you have to remember that the oxygen, it is transported from the alveoli to the tissue by the blood in two forms, as a simple solution or in combination with the hemoglobin. So for entrance exam, you have to remember that transport of oxygen is maximum is in combination with the hemoglobin. Around 97% of oxygen it is transported as the form of oxyhemoglobin. So the oxygen, it will combine with the hemoglobin that is present in the blood, and it is transported as the oxyhemoglobin. So it is 97%, whereas 3% it is the oxygen, it dissolves in the water of the plasma and it is transported in this physical form. The next important thing is the oxygen hemoglobin dissociation curve, also known as oxyhemoglobin dissociation curve or the oxygen dissociation curve. So this curve, it is an important tool for us to understand how our blood carries and releases the oxygen. So the oxygen hemoglobin dissociation curve, it relates the oxygen saturation and the partial pressure of the oxygen in the blood. And that is why it is determined by the hemoglobin affinity for the oxygen. So that is how readily the hemoglobin acquires and releases oxygen molecules into the fluid that surrounds it. So as we know all that hemoglobin, it is the primary vehicle for the transport of oxygen in the blood. The hemoglobin molecule has capacity to carry 4 oxygen molecules. So these molecules of oxygen will bind to the iron of the prosthetic group. So the oxygen bound to hemoglobin, it is released into the blood plasma and it is absorbed by the tissues bound to the hemoglobin. So the saturation of the hemoglobin with the oxygen, that will depend upon the partial pressure of the oxygen. When the partial pressure of oxygen is more, then the hemoglobin will start accept oxygen. And when the partial pressure of oxygen it is less, the hemoglobin it will release oxygen. So what is the shape of the oxygen hemoglobin dissociation curve? You can get a question in your exam. Under normal condition, the oxygen hemoglobin dissociation curve is S-shaped or the sigmoid shape. The lower part of the curve indicates the dissociation of oxygen from the hemoglobin, and the upper part of the curve indicates the uptake of oxygen by hemoglobin, depending upon the partial pressure of the oxygen. So the oxygen dissociation curve has a horizontal axis and a vertical axis. The horizontal axis is amount of the oxygen available, and the vertical axis is the amount of the hemoglobin that is saturated with the blood.
So let us discuss what happens to the oxygen dissociation curve when it shifts to the left. A shift to the left means there is increased oxygen affinity. So that means there is increased affinity means there is tighter binding due to the higher oxygen saturation in relation to the partial pressure of oxygen. And what happens during shift to the right? That means there is decreased oxygen affinity. So there will be the oxygen will be loose bind, and hence there will be release of oxygen to the tissues. In what conditions the oxygen hemoglobin dissociation curve shift toward the right? So decrease in the partial pressure of the oxygen, increase in the partial pressure of the carbon dioxide, increase hydrogen ions, and a decrease in the pH, increase body temperature, and excess of 2,3-diphosphoglycerates in RBC. These are the condition when the oxygen dissociation curve, when the oxygen hemoglobin dissociation curve is shifted to the right. The oxygen dissociation curve is shifted to left in the fetal blood because the fetal hemoglobin, it has got more affinity for oxygen than the adult hemoglobin, or when there is decrease in the hydrogen ions and increase in the pH. What is Bohr effect? Bohr effect is the effect by which the presence of carbon dioxide, it decreases the binding of the hemoglobin, it decreases the affinity of the hemoglobin for the oxygen. In the tissues, the partial pressure of the carbon dioxide is very high and the partial pressure of oxygen, it is very low. So due to this pressure gradient, the carbon dioxide will enter the blood and the oxygen is relieved from the blood to the tissues. So the presence of carbon dioxide will decrease the affinity for hemoglobin for the oxygen, that will enhance the release of oxygen to the tissues. And if the oxygen that will cause the oxygen dissociation curve to move toward the right. So all the factors that shift the oxygen dissociation curve to right will enhance the Bohr effect. So what does the right shift of the oxygen dissociation curve indicate? Just remember, a right shift indicates when there is decrease oxygen affinity. So if there is decreased oxygen affinity for hemoglobin, the blood will release more oxygen more readily. So what is the difference between the Bohr effect and Haldane effect? The Bohr effect is the decrease of the hemoglobin oxygen binding capacity with the increase in the carbon dioxide concentration or decrease in pH. While the Haldane effect is the decrease of the hemoglobin carbon dioxide binding capacity with increase in the oxygen concentration. So the Bohr effect helps the metabolizing tissue release oxygen from the oxyhemoglobin, but the Haldane effect helps the lungs release carbon dioxide from carboxyhemoglobin. So the Bohr effect is important because it improves the oxygen supply to the muscles and the tissues where the carbon dioxide production occur. So that aids in delivery of oxygen to the area where it is most needed. So is the Bohr effect good? Yes, the Bohr effect allows for the better oxygen unloading in the metabolic active peripheral tissues like the skeletal muscles during the exercise. So increase skeletal muscle activity causes the localized increase in the carbon dioxide partial pressure that lower the local blood pH. So why does the pH decrease when the carbon dioxide increases? So that happens because the carbon dioxide, it reacts with the water to form carbonic acid, and the carbonic acid dissociate to release hydrogen and bicarbonate. That lowers the pH and the pH will drop as a result of this. Now transport of the carbon dioxide. The carbon dioxide, it is transported in blood in four ways: as dissolved form, as carbonic acid, as bicarbonate form, and the carbono-amino compounds. So the maximum is through the bicarbonate, that is 63%, that is transported as bicarbonate form. So from the plasma, carbon dioxide, it will enter the RBC. In RBC, the carbon dioxide combines with water to form carbonic acid. The carbonic acid is very unstable. It dissociates into bicarbonate and hydrogen ions. What is chloride shift? It occurs when the carbon dioxide, it enters the blood from the tissues. In the plasma, plenty of sodium chloride is present that dissociate into sodium and chloride. So when the negative charge bicarbonate ion that moves out of RBC into plasma, the negative charge chloride ions moves into the RBC in order to maintain the equilibrium. So that is the chloride shift or the Hamburg shift. So it is the exchange of the chloride ion for the bicarbonate ion across the RBC membrane.